with the Chemometic webinar. Welcome. The webinar is the meticulous researcher's guide to validating your cell counting method. My name is Sanneke Cotier. I'm based in Bristol, the UK, and I'm part of the customer support team in the UK and Ireland. I will be your host today, which means that I will looking at question and answer sessions later on, so after the presentation. With me, I've got my colleague Annemieke Karyotis, so she will be doing the presentation. Annemieke is one of our field application scientists, and our field application scientists do all the technical support for our customers. Annemiek's got a PhD from the University of Edinburgh, where she looked at apoptosis and neutrophils, and she has many years of technical support experience. So starting with fluorescent imaging and Dynol, which shows the many years, cell separation, but after that mostly flow cytometry. And we were lucky to get her a couple of years ago. So now she's been for several years with Chemometic, advising on cell counting and how to get the best cell count you can get. She's seen all sorts of samples and cells that are difficult to count, and yeah, she helped with all of those. So before I begin, a little bit of housekeeping. You might have seen that we don't have the chat open. There are too many of you to deal with any questions during the presentation. So if you can put that in, there's a question and answer box at the bottom of your screen. So if you can put your questions in there, then we can collect them. The presentation is about 20-25 minutes. And after that, we'll have 10 minutes of question and answer, and we'll try and answer as many questions as possible. So I will leave it to Annemieke to start the presentation. Over to you.
Okay, thank you very much, San. Right, I'll just share my screen. Okay, well, thank you very much for joining me this afternoon. The reason we wanted to cover this particular topic in terms of validating cell counting method is because it's one of the most common questions we get at Chemometic. So without further ado, and I will just click here, there. So one of the key takeaways we'd like to take you through this afternoon is talking about validation of the cell counting method. Although all of the data I'm taking you through is actually with our newest instrument, the NC202, the experiments and the theory behind it is applicable to any cell counting method. So it should be applicable to any of you. So we're going to go through accuracy and precision of cell counting methods, specificity, reproducibility, and different effects of method variability, and all these different considerations that might be important in, say, your cell manufacturing process or your antibody production process. And as kind of some of the experiments are relevant to different processes or manufacturing processes, but not all. Firstly, I would like to just introduce Chemometic. Chemometic's been around for many years now, and we have thousands of customers across the biotech and pharma and academic segments. And based in Denmark, we aim to produce high quality, precise instrumentation to our customers. So a little bit first about the language of cell counting and what the different terms mean. And a lot of this presentation is based on the ISO Part 1 and Part 2 guidance, general guidance on cell counting methods. And we're going to cover probably the most common questions that we get. And a lot of the common questions that we get are things regarding, I have two different cell counting methods, and I get two different values, which one is right? And so how do you define that? So accuracy refers to how close is my result to the true count of the measurand. Precision refers to how repeatable the results are that I'm getting. And so they're slightly different things. Specificity is your cell counting method's ability to cope with any other analytes that might be present, like a contaminant or a magnetic bead. And then ruggedness, if there might be effects, small effects on your cell counting method, say time of day, different temperatures, this sort of thing. So we'll start with accuracy and precision. Accuracy refers to how, if you look at the first, the gray diagram, this is the ideal cell count. So accuracy is where you're hitting the bullseye. You're just as true as you can to the actual theoretical true count. So you're actually very accurate. Precision, if you look at the yellow diagram, is you're very precise and you're very repeatable in your results, your cell count, but you're not accurate because you're not hitting the bullseye. And then you have the blue diagram, which is an unacceptable cell count, because you're neither precise nor accurate. This paper came out, I think, about a year ago now, International ISO Standard Part 2, and it was very good for coming up with a method to look at accuracy, because in cell counting, we don't have a reference method or a reference standard where we can compare what is the actual count. So you have two different methods. Who is right? How do you understand that? Because even if we had a reference method, it would still have to be counted. And how do we know that that count is actually true, the true value? So they came up with precision and the principle of proportionality. And to achieve a high confidence in cell counting, it means that you have to be both accurate and precise in any cell count. So if you have an accurate cell count, the hypothesis is, you should be able to take a top concentration, dilute down, and come with a perfectly straight line. And they sort of refer that this should be an indirect measure of accuracy in a given cell count. So the difference between accuracy and precision is accuracy is going to be the proportionality of a serial dilution, and precision refers to the percent CV of replicate measurements. Accuracy. So here we took the NC202 and measured using the precision of the principle of proportionality. So you take a top concentration of cells and do doubling dilutions down and check for linearity. And here you can see the NC202 has showed great linearity. This was done on adherent CHO cells. And the other thing that I just want to point out, that although we've got excellent linearity here, and you can, in many experiments where you get excellent linearity, it's really important to keep a really good eye on the precision. So your percent CVs of each data point. And we have seen examples where you have good linearity, but the CVs are not acceptable. And I think you'll know who I'm talking about if you're listening. So the other thing that's really important to look for is linearity of viability. That also should be linear, of course. So in this experiment, we took live cells, and it has to be really good live sample of cells, say at least 95% or above, and then mixed it with dead cells and in a known ratio. And here we go. So again, the NC202 gave great linearity when we mixed a dead to live cell ratio with viability. We freeze shocked these CHO cells. You can also use, for example, ethanol to kill your cells. There's various methods. So what we looked at now in the past few slides was immediate accuracy and precision. What we would also really want to test for is intermediate precision. So this really refers to how your cell counts or your CVs behave over time. So we grew some cells in a flask over time, and then took triplicate samples for each day to check that these CVs are within acceptable limits. So here we go. These are HEC cells grown for about 16 days. And each day we took triplicate samples. And you can see the yellow line is where this total cell count is increasing up to the top. And then as they run out of food, they start to die. And with that, you got nice viability data where the viability is high. And then around between day 9 and 10, it starts to decrease. The important thing is that the CVs are all within acceptable limits over time. So then we come to method validation and verification again. But then looking at other parameters like inter-operator and inter-device variability, if there's an unacceptable source of variation with these parameters. And inter and intra-operator variability is probably one of the most important parameters that you need to check for in terms of cell counting, because there's a huge variation in skill and ability between operators in terms of how they handle the samples and what CV they're going to end up with. So you might, for example, get a very good CV when you perform a cell count, but your colleague doesn't get acceptable CVs. So your cell counting method has to be robust enough so that there's not too much variation between users. And actually, as a side note, we commonly see this where there's high CVs, and the reason is because the most important part of cell handling when you're doing cell counting is the prior mixing of the sample. And it should be the prior handling and mixing of the sample should be the same between all operators. So if, for example, operator A pipettes up and down 5 times immediately prior to sampling, then all operators should do exactly the same. And in fact, many of our customers have this written into their SOPs, how the sample should be handled, because cells settle so fast, and then you end up with poor CVs. So in this experiment, we gave each operator 5 minutes training. They hadn't used a nuclear counter before. 5 minutes training, and then asked them to count the same sample, each of them 6 times. And we were checking that their individual CVs were acceptable, but also that they got the same total cell count as each other. And here we were able to see that there was no statistical difference between the operators. So the cell counting method here is robust enough to deal with different operators. So I'm going to ask you if you'd please do a poll for me, just because I'm really interested to know how all of you do cell counting and all the different, couple of other things. So now I need to find the poll button. And basically just because I'm a bit nosy, I would like to know. So if you do, I'll give you 30 seconds to answer the questions. It's just 3 questions, it's really fast. So it's, how do you currently count your cells? What percentage of time does your group spend on cell counting? And how important is accurate cell counting to you? Okay. Getting some answers. Let's see, it'll be interesting. So I'm going to share the results at the end of the webinar, so that you can see also. It'd be maybe quite interesting to see about each other what the answers are. So I've left a bit longer than 30 seconds, so I think that's it. So I'll end the polling now. Okay, and then I will share the results at the end. Thank you very much for doing that. Okay. So the other thing you might look for in terms of testing your cell counting method would be looking at instrument to instrument variation. So you might have, for example, 3 different cell counters across a site or even on the other side of the world. But the important thing to know is that they all count exactly the same so that you can share protocols. So in this experiment, we took the same sample, of course, split it into separate tubes, and then counted the same sample on 5 different NC202s. And what we found was that we had good inter-instrument consistency in terms of total cell count, and then also inter-instrument consistency across the viability. Specificity. This is probably another really important parameter for some of you in, say with cell production or cell therapy. If you look at your process across the whole, each time you're doing cell counts, is there anything new that's been introduced to the sample? For example, magnetic beads. Has there been a magnetic bead step? Could you possibly have red blood cell contamination? This kind of thing. So in our case, we got some PBMCs and we got some purified red blood cells. And we took the purified red blood cell, the PBMCs, and added in red blood cells in increasing concentrations, and then looked at the counts to see if it affected the counts, and 2, if it had an effect also on another machine as well that we looked at. So what we looked at was the 202, which is the blue bars, and another instrument that uses bright field microscopy, and to see the effect of the red blood cell contamination. So even up to, I think it was the top concentration 2 times 10 to the 7 red blood cells per mil, the NC202 was not affected, the cell counts were not affected. However, you could see that quite the other instrument was quite badly affected in terms of cell count. So it's important to look at these things and see if the cell counts are affected. Okay, so I think that's the end. We've come to the end of the webinar. But I would promise to share the poll with you. So let's have a look. Share results. So let's have a look. What have we got? How do you currently, so we've got 21% is manual, 79% automated. That's interesting. What percentage of time does your group spend on cell counting? The majority, less than 10%, 10 to 20%, 20 to 35%, and 3%, almost 100%. That's quite a lot of you actually spending quite a significant amount of time doing cell counting. And how important is accurate cell counting? Yeah, it's important, very, very important to most of you. Okay, well, that was very interesting. Thank you so much. Right.
Okay, over to the answering questions. We've got a few questions coming in already. So
okay.
Are you ready, Annemieke?
Just let me stop sharing. Right. Oh, stop share. There we go.
The first question is, and thank you for your questions. It's really good to get these questions and to share your experience with cell counting. So first question is, do you see different levels of accuracy precision depending on cell types? And it says, we manufacture cells from donor monocytes and we see poor accuracy when viability starts to drop off, say below 70% viability.
Oh, well, yes. I mean, we do see lots of different reasons and for getting poor accuracy, but it can be a million reasons for each different scenario. For example, we've had situations where the counting was performed in PBS, and that affected it because you lost all your cells. They stuck to the sides, and you know that ended up in poor accuracy. So yes, but if I'm happy to also, we'd have to have a look in depth as to what the whole process was, and then investigate as to what the reason is. So I can't really answer without having all the details, but yes, we do see lots of different things.
Yes, and we can also contact you and just go in more detail on that.
Of course, yes.
There is another question, and this is an interesting one actually, because it says the NC200 and the NC202 count cell nuclei.
Mm-hmm.
Therefore, you don't see an effect with NC. I think that's non-nucleated cells. Mm-hmm. But yes, can you answer that about the limitations of counting with cell nuclei?
Oh, well, you mean that because the RBCs are non-nucleated that we won't see an effect?
Yeah.
Oh yeah, but we do eventually, because the I think it's the hemoglobin, it quenches the acridine orange fluorescence.
Okay. I hope we answered that correctly, but if we haven't, we'll contact you separately. Somebody asks, how often do you need to calibrate the machine?
Well, you don't really need to calibrate the machine per se. You'll know when something is wrong. The thing is more from a paperwork and GMP point of view, because we have customers calibrating their machines once every 6 months and sometimes once a year. It really depends on what you need in terms of paperwork.
Okay, thank you. And here is a question: are the findings shown regarding consistency applicable to other instruments in the chemometric range? And have you tested consistency differences between your different instruments? It says here, for example, the 202 against the NC250.
Yes, so we've done all these tests on all our instruments. So we have the data in-house.
Yes, I add to that as well that even if we tested it, it's different with different cell types. So we're happy to test that with you.
Yes.
Yes. So let me see. Oh, we've got more questions. We still have time for a couple more. So I noticed that the PBMCs always do not fall within the gating when using the NC200, and we had to adjust the gating accurately. So we've got quite a lot of customers here, I could see.
Yeah.
So with the NC202, you have a different strategy. And how do you ensure that the gating strategy is accurate with the 202?
So the 202's got a slightly better camera in it than the 200, and so you get a better dynamic range, and so you're better able to separate debris from your main cell population.
Yes, good. We've got actually quite a lot of questions, and I don't know if I've got time to go through them all. I'll do another 3. And then if we need to, we can always email you back. It says here, is there a way to minimize human error in cell sample dilution?
Yes, and in fact, thank you for asking that question. So when you come to do these linearity studies, please do get in touch, because we have there's quite a lot of tips and tricks involved. Like, for example, don't ever dilute in PBS. There's a few different things that you should be aware of, like, because if you dilute in PBS, you lose your cells to the plastic on the sides. So before you go and do linearity study, please get in touch, and we'll tell you all the different tips and tricks for that.
Okay. I'll do another 2. It says here, how does the NC202 compare to methods such as the tripe and blue method? And what reference do you use to calibrate the NC202?
What reference do we use? Yeah. I'm not quite sure I understand the question.
Well, I think it's actually, the thing that you showed in the beginning, because there is no reference, right?
Yes, exactly. That's yeah. There's no reference. And so basically, for each method, you would want to do a linearity study, and make sure that your CVs are acceptable for whatever method you're using.
Yes, you've got so many questions. I'm trying to read them really quickly to see, you know, which ones we can discuss that haven't been repeated. So we thank you very much for all your questions. Yes, I've got one more question that might be useful. And that is about triplicates. So you mentioned that you were using triplicates
Yes.
in your counting, and here it says people see that in different ways. So what do you mean by when you use triplicates?
Oh, right. Yes. So it's all about looking at, so it's always this counting the same sample in triplicate, because we're trying to understand what the variation is in the process, in the operator, in the machine, all these different sources of variation, but we want to keep like the same sample, the same instrument, so that we understand what is the natural error of that cell count method.
Yes, good. Well, as I said, lots of questions came in at the very end, so you know, great to have all your questions.
But we can answer them, right, afterwards?
Yes, so we will make sure that we'll get back to you, because I can see the questions, and we'll make sure that we get back to you with all the questions that you've asked. I'd like to thank you for joining the webinar. I hope you found it interesting and that it will help you with your cell counting. We're here to support you, and feel free to contact us if you want to use the expertise of our technical specialists, who all they do is do cell counting. So we're happy to help you with any questions that you have.
Yes, please do get in touch, and we will answer all the questions.
Good. Thank you.
Thank you for joining.